Right now, somewhere in the world, a child is being born with a disease written into their DNA, not caught by virus, not caused by lifestyle, just written there. You and I live in a world where some things are accepted as they are. Your genes are your genes, either they work or they doesn't. Game over before the game started. For many years, this has been the baseline of biology and human body. But quietly, in the recent years, something has changed, and that's why you need to know about it. DNA is the first problem, protein structure are the second one. It took 12 years for a scientist named John Kendrick to map out a structure of a protein from the heart of a whale. 12 years for one single protein structure, and he won Nobel Prize for it. And over the years, after the invention of John Kendrick, 150,000 protein structures were invented in 60 years. 60 years for 150,000 protein structures. But a company has done insane thing of mapping out 200 million protein structures, almost all of the proteins known to humanity and that are present in the world in just 1 year. And this is another insane technology. And there are two technologies which I am going to speak about in this video. The first one has edited a gene, and second one has mapped 200 million protein structures in 1 year. These two technologies suggest something that sounds insane. Life might be becoming programmable, not metaphorically, literally. These are the two main pillars that are the main topics of this video. Pillar 1 is about reading biology, understanding the machinery of life like never before. And pillar 2 is about writing biology, understanding that machinery and editing that machinery like never before. So in this video, we are going to understand these two pillars one by one. Pillar 1 is AlphaFold, learning to read proteins. First, you need to understand what proteins are. Your body is made up of 20,000 chains of amino acids, like a beads on a string. All these proteins are the workers of your body. They carry oxygen, they fight infections, there does a lot of work. And these coils on a string, they match up, they react with each other, and they push, pull and coil together to form a complex 3D structure that has been ununderstandable for many centuries. This shape is unpredictable, specific and intricate, and through this we cannot understand the protein itself. So this was the protein folding problem. This protein was the main cause of diseases like Alzheimer's, Leishman's, Parkinson's, Chagas and malaria. These diseases were caused because of the miscoiled or misunderstood proteins. You basically cannot design a drug for a protein you could not see. So we were blind for so many years. Team in 2020 at Google DeepMind has built something called AlphaFold 2, an AI tool learned through deep learning, and all the protein structure that has been mapped by the scientists from all over the world has predicted the structures of proteins accurately, almost instantly, at a scale no human could achieve. COVID-19, AlphaFold predicted the structures of SARS-CoV-2 proteins, giving a faster way towards vaccine research. AlphaFold is also imperfect because it stumbles while producing our structures of antibodies. We have learned to read biology, imperfectly but better than ever. Pillar 2 is CRISPR, learning to write. CRISPR started as a discovery, not as an invention. Scientists found out that bacteria had natural immune system. They store the snippets of the virus that came and attack the bacteria. So they designed a molecular scissors called Cas9 that act as a scissors to cut out the exact snippet in that bacteria. Scientists Jennifer Doudna and Emmanuel Charpentier designed a molecular RNA that could match to any DNA sequence, and they also designed a molecule called Cas9. Cas9 would follow the molecular RNA and acts as a scissor. Victoria Gray was born with sickle cell disease. Her red blood cells, instead of being round and flexible, they were sickle shaped. So she had to spend months in hospitals. So doctor took the blood cells from her bone marrow, edited it using CRISPR and put billions and billions of cells back into her bone marrow. So they started to produce fetal hemoglobin. She was cured from a disease which was considered incurable. Baby KJ was born in 2024 with a CPS1 deficiency, a rare metabolic disorder. So while being an infant, he has to spend more and more time in hospital and in permanent danger. So scientists at Children's Hospital in Philadelphia and also the students at University of Pennsylvania joined together and designed a specific genetic therapy for a single individual in 6 months. They cured a mutation in his liver cells. He was cured from any potential danger, and he's growing and well thriving. Personalized genetic therapy designed and delivered in months for a single individual. Just understand the significance of that sentence. For all of human history, biology was just a territory. We could adapt, we could treat symptoms, and we could learn from it. That's it. The code itself was untouchable. With these two technologies, AlphaFold helps us to read the code. CRISPR helps us to write the code. So we are transforming from observing life to potentially controlling it. This is not any type of technology. It is a categorical change. You may be wondering why this matters to you. Because decisions about this revolutionary technology is not happening 50 years in the next future. No, they are happening right now in the hospitals, in the boardrooms, in the government meetings. So that's why it matters to you. Let me be honest with you. This is not any good news story. In 2018, a Chinese scientist named He Jiankyu used CRISPR to edit human embryos. Two twins named Lulu and Nana were born with editable DNA. These DNA were edited to give resistance to HIV. International community reacted with horror. These edits were made in secret without any proper consent. These girls may have received unintended changes, and these are heritable edits, means they could transform from one generation to another generation forever. But CRISPR has two main loopholes: off-target edits. This is one of the most asked questions. What if CRISPR edits a wrong gene? And the second one is access. Victoria Gray's treatment almost took $2 million, and KJ's was research funded. These technologies exist, but they are highly expensive, and there is a deeper question that nobody has answered. Where is the line between good use of CRISPR and bad use of CRISPR? CRISPR has already been used in pain tolerance, mass increase, and also to modify sleep cycles. Where is the line between treatment and enhancement? In Jennifer Doudna's own words, that is a defining, challenging moment. So you came into this video knowing that diseases were as good as fate and biology is something out of control. You may not have heard about AlphaFold. You may have heard about CRISPR, but you would not have known what it does. Now we know that one AI has predicted the structures of 200 million proteins, opening doors to many incurable diseases. And now also you know that woman who has been spending her life in hospitals is now living free of sickle cell disease. That another baby has been cured from CPS1 deficiency because of a specific genetic therapy designed for that baby in 6 months. And then also you know that these revolutionary technologies could be misused and have been misused. So we need to make decisions and we need not to be ignorant because we cannot afford the consequences. We started this video in a world where biology was something that happened to us. We're ending this in a world where we are learning to edit biology. This is an enormous power. The decision of how you use it is not done by the technology itself. It is done by the people who use the technology. Jennifer Doudna said it better than anyone could.
Few technologies are inherently good or bad. What matters is how we use them. And when it comes to CRISPR, the possibilities of this new technology, good and bad, are limited only by our imaginations. I firmly believe we can use it for the former and not the latter, but I'm also cognizant that this will require determination from us, individually and collectively. As a species, we have never done anything like this before. But then again, we've never had the tools to do it. The power to control our species' genetic future is awesome and terrifying. Deciding how to handle it may be the biggest challenge we have ever faced. I believe, that we're up to the task.